Let me give you an introduction, then I will go over some of the things in your book. So DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. It's because of the kind of sugar that is present. Both of them have a sugar. Each of the DNA and RNA is made of a basic unit called the nucleotide. Like, what was the basic unit for sugars? Glucose. And for proteins? And for lipids? Fatty acids. The same way for DNA, RNA is the nucleotide. So every nucleotide will have a sugar. There will be a nitrogen containing compound, they call it nitrogenous compound, and a phosphate attached. Okay, the sugar, nitrogenous compound, and the phosphate. The sugar is ribose, one kind of sugar. In this case, that sugar lacks one oxygen, so they call it deoxyribose. This is the normal ribose. That's the one basic difference between the DNA and RNA in terms of the sugar. And these nitrogenous compounds, as you can see, the DNA structure and function at the bottom of the page, the nitrogenous compound, they also call base, nitrogenous base. You have the A, T, C, G highlighted, right? A stands for adenine, C for cytosine, T for thymine, and G for guanine. But in case of RNA, the guanine will be replaced by uracil. So you will have adenine, thymine, guanine, cytosine. So these are the four of the possible nitrogenous bases for DNA. In case of RNA, this will be replaced by uracil. So in short, they use the notation A, T, C, G. Within parentheses, you can write U, meaning in RNA you won't find the T's. Okay, and there is also some rule by nature because of the structural affinity and the structural configuration. Only A can pair with T and C can pair with G. This is a rule. They call this complementary base pairing. They complement each other. Base pairing because they're all the nitrogenous bases. And because of the structural configuration, again, the way A can match only with T or the other way around, or C with G. So that's what gives the DNA the winding staircase model or appearance. Okay, so you'll see the steps of the winding staircase and the railings, as you will see in your book in the next page. Okay, so you'll have your A's, T's, C's, and G's. So you'll never find the A and the G or C and the T. You find A here on the opposite side, you'll find only a T. You find a C, you'll have a G. If you find a G, you'll have a C. Okay. If it was RNA, first of all, RNA is not double-stranded. RNA is usually single-stranded, and you'll find there will be no T. Wherever a T should be there, you will find a U. But there are some RNAs which have a shape like this, like flower-shaped. So if you have a shape like that, so there may be some regions where you'll find some double strand, but generally it is single-stranded. So you'll find the double-strand portion of the RNA double strand in tRNA. So coming to that, the RNA is of three kinds. You have messenger RNA, that comes from the template, the DNA template. There are enzymes that make this messenger RNA, and your ribosome itself has an RNA, that is ribosomal RNA, because it's part of your ribosome. And the third category of RNA is tRNA, which is called transfer RNA, T for transfer RNA. So the M stands for messenger, because it serves as a messenger between your nucleus and the rest of the cell in terms of carrying the genetic code and the DNA sequence to the cytosol. Then you have the ribosomes, which have the rRNA that help to recognize this messenger RNA and bind. Once they bind, then you go to the ER, okay? And then there is this tRNA floating around in the cytosol. They all carry a specific amino acid. This nucleotide sequence corresponds to the mRNA sequence, which corresponds to the DNA sequence. So everything is predetermined in the DNA sequence, and the mRNA sequence will be complementary to that. And based on the mRNA sequence, the tRNA with whatever amino acid will come and attach here, and the first amino acid will be in place. Like that, you will add more amino acids to that. We'll see that in a couple of minutes.
For the second one, the underline, is that a lowercase r?
Yeah, all starts with a lowercase. Okay, now let's go to the book with that short introduction. So you can see on figure 4.1 and 4.2, you can see the nitrogenous bases and the standard structure of a nucleotide. On the top, you see the adenine, the deoxyribose, and the phosphate. Okay, I did write on the board each nucleotide has a sugar, phosphate, and a nitrogenous base, right? So if it is the adenine nucleotide, you will see adenine. Depending on which one we are talking about, you will have a different nitrogenous base. It could be A, G, T, or C. The adenine and guanine, they call it purines, and the other three pyrimidines. Then you can see in figure 4.2 how the DNA structure is made. So you can see the complementarity between the A and the T and the C's and the G's. It could be either way, but it has to be either of one of the two. A can pair with only T, or the other way around. C can pair with G, or the other way around.
It doesn't even change any mutations in DNA?
Yeah, if it happens, usually there are enzymes that keep proofreading all the time, and they fix it. Very rarely mutations occur. It's in only extreme cases mutations occur. Mutations may occur, but it's fixed. Even if mutation occurs, like sometimes for the amino acid, there is more than one 3-letter code. There are some possible options to make the same amino acid. So if the mutation doesn't change the meaning in terms of amino acid, nothing happens. So there are several checks and balances for the cell. But that's something we'll see in the chapter as we move further. So they have talked about the base pairs and the law of complementary base pairing in the next page, 117. And you can see the guy who proposed the structure for the DNA with his colleague Watson, James Watson, and Crick, the other guy. But the lady in the picture on the top, she was the one who was basically involved. But these guys did more work and completed the work and got the credit, as it happens sometimes, most of the times. Figure 4.4, next page. I was talking to you about the different levels of structure formation. So what do we have from step 1 through 6? That's what I was talking about earlier. What is chromosome, DNA, chromatin, and all of that. So that picture tells you the story. So on the top, you see the DNA sequence, the 2 strands. In step 1, the double helix. Then you wind the DNA around some proteins we call histones. Histones are the kind of proteins in the picture. It says core particle, but if you see above the picture, you see the term highlighted histones. Histones are the proteins around which the DNA is wound. And once it's wound, then it goes through further folding, and finally, if you see, it's more condensed, and now we are going to call it chromatid. So is it the same thing, structural changes, different names. So if you take the individual stretch of the chromosome and let it look out, that's your DNA sequence. But when you wind it, it goes through more structural changes. Then with the proteins, it's called chromatin, and then when it's more condensed, the final structure, you call it chromatids. Chromosomes, normally we use chromosomes, but during cell division, we call them chromatids. So that's why you see in dividing cells only the last one, they are boxed, and the top there is a caption. In dividing cells, we call them chromatids. And the chromatids are attached together in the middle by a structure. Centromere is in the center of the tube. That's where when your cells divide, the fibers will come, microtubules will form some fibers, they'll come and attach and then split the chromatids apart. Under RNA structure and function, they have discussed what I already explained to you, the three types of RNA, the messenger, ribosomal, transfer RNA, they're all italicized. And what is very unique about RNA? One thing, the sugar is different. Instead of deoxyribose, it's ribose. And the other difference is single-stranded, not double-stranded in most cases, with exception. And then the nitrogenous base T is replaced by uracil. Again, I have a few videos on my homepage, and also you can type some of these terms if you need help on YouTube, so you can look up some animations. So the next aspect is the gene, genome, genetic code. Okay. Before you go into that, I want you to understand the basic fact of life. DNA always needs to copy itself, make more copies, identical copies. We call it, in simple terms, duplication, but we call it DNA replication. You have a DNA molecule which has to make another copy. This happens during cell division. So if you did the lab already, you would have talked about the cell cycle and the cell division. During the cell cycle, there is a phase called S phase where it makes copies so that it can divide the chromosomes with the daughter cells. And then on the same cell, you also make mRNA and protein, so that when the cells grow and do their routine activities, so you have these steps we call transcription and translation. So DNA keeps replicating itself as the cells divide, and when the cells keep doing their specific jobs, they go through these steps, the transcription and translation. So that's the fundamentals for life and living organisms as such. Okay. But we will see how these things work. So what is a gene? So any sequence of DNA, those nucleotide sequences that corresponds finally to a protein. It's called a gene. So you can have a gene for your eye shape, eye color, the iris color, the nose shape, or there may be more than one gene for the same phenotype, the characteristic that you are looking at from the outside. So they call genotype and phenotype. Genotype is the gene or genetic makeup. Phenotype is what is being expressed on the outside, what you see as skin color, hair color, eye color, and so on. Okay? So the genes are the sequences of DNA that contain the information to make proteins. So in the book it says information containing segment of DNA that codes for the production of molecule of mRNA, which in turn helps with the synthesis of proteins. So what is the genome? Genome is for the whole organism. Like human genome has like so many genes. If you take a bacteria, there will be fewer genes. Okay, the higher the complexity of the organism, you'll find more number of genes for different structures and different activities.
Your genes can change, right? Because, like...
Yeah, they change. That's how we look like what we are from a monkey or a whatever our previous ancestors looked like.
Yeah, I was going to say, because like in pictures of myself when I was a baby, I had blue eyes, and a few months later...
It doesn't change that fast, by the way. It takes millions of years, unless there was some mutation. Unless there was some mutation.
Well, I didn't hear what you had said.
I said it takes millions of years to change, unless there was a sudden mutation or something, you got exposed to something, I don't know. But normally it doesn't.
Yeah, normally it doesn't.
So, let's look at the genetic code. They have talked about genomic medicine and all. We don't have to worry about that now. That just says that you can play with genes these days, like you can treat some of the diseases and disorders with gene therapy, but it's still at the very developmental stage. It has not gone too far. So genetic code is contained in the DNA sequence. Okay. So they are always read in three nucleotides. It could be A, T, C, A, C, T, whatever. Then in your mRNA that is being made based on what you have here and following the rules. Remember, and you are making the messenger RNA, it's an RNA. So wherever T goes, it has to be replaced by U. So meaning if you had a ATC here, you will have a U, because there won't be a T in the RNA. A, G, it will be again a U, G, A, and so on. Okay. So whatever the triplets, three nucleotides you have in the DNA, they call it genetic code. The same triplet that corresponds to that, you will call it codon. And then you will have a tRNA molecule that will have something complementary to that. Again, it's an RNA, so you have to have complementarity. So it should be AUC. So this three letters will correspond to a certain amino acid. So as you can see, whatever you have, the sequences, they drive the whole process, your transcription and the translation to the protein. Okay, so let's look up some pictures you have in the book so you can understand better. In this triplet they call anti-codon because it's opposite or complementary to the codon. The genetic code is in the DNA, codon is in the mRNA, anticodon is in the tRNA. So under genetic code, you will see all these discussed, the base triplet, codon. And also you have codons to tell where to start, where to stop. The stop codons Normally the start codon is going to be an AUG, meaning you should have here T, A, C to start with. And the stop codons can be any of these. So start codon is always AUG. The stop codon is one of these. It's where my biology, basic biology 1005 class, I tell them these to remember, but it helps at any level, I guess, how to remember which codons are stop codons.
What is the first one?
You are gone, you are away, you go away. Meaning, it's telling the ribosomes to stop making the proteins. But you don't need to worry about that if you can remember this. So you need to know what is your start codon and stop codon. And table 4.2, you can see some examples of the genetic code. So first you have the DNA triplets. You see the corresponding codons of mRNA and what they represent, which amino acid. So if you have the first one, if you see table 4.2, you have CCT. So if you have CCT here, let us say your mRNA will have GGA. Okay, that GGA is the codon that codes for amino acid. So that codon will code for glycine, one of the 20 amino acids. There are only 20 amino acids. There are 4 bases, possible bases. So in combination, they make up to 64 codons. You have 4 bases, right? The AT, CG. If it is RNA, it's AU, CG. So your message is always read in threes. So the possible options are 4 times 4 times 4, meaning you will have 64 total codons that will correspond to 20 amino acids. So what does that mean? You have 20 amino acids, but you have 64 codons. There are some amino acids that have more than one codon. So to summarize, if you want to look up figure 4.6, you can see the steps. First you see the gene, the DNA sequence that goes through transcription, and then you form a messenger RNA. But if it is a higher level organism like some of the animals, humans and plants, they go through a pre-mRNA step. So if you see the pre-mRNA, there are some dark pink and light pink regions labeled A, B, C, D. The dark colored regions are unwanted regions, meaning they're not necessary to make the proteins. So they are all cut, or they are chopped off by the enzymes that do this inside the cell. And then you bring the A, B, C or A, C, D, whichever you need together. So from one stretch of your DNA sequence, you can see the mRNA can be combined in 3 different ways. Can you see that? You see the pre-mRNA and mRNA, see what is happening there. Okay, they call it gene splicing, meaning you bring the different regions of the genes together and match in different ways, and then you make your mRNA in 3 possible ways, or it could be more. It's just they have given you some examples. So the one stretch of your mRNA can be brought together in different ways to make 3 different proteins. So you can see protein 1, 2, and 3 right at the bottom. So this shows you the various steps involved in transcription, translation in one figure, but you have more details in the following pages. Figure 4.7 shows your transfer RNA. like I said, parts of the transfer RNA are double-stranded, so you can see that there. See the link in some places from both sides. It's the same piece, but it's arranged in a way that it needs more stability, so you have a partial double strand. Usually they are single-stranded. Then under transcription, they have described how the transcription occurs based on the nucleotide complementarity, which I already talked about. And the enzyme that does this is RNA polymerase, because it's helping to make the RNA, mRNA, polymer of mRNA. Then translation, under translation you find the different types of RNA, messenger RNA, transfer RNA. The triplet in the messenger RNA, we will call it the codon. The triplet on the tRNA, we call it anticodon. Then you have the ribosomes. Ribosomes, each ribosome has two subunits, one small and one large subunit. And there are sites where the tRNA with the amino acid will come and bind. So your messenger RNA is here, the ribosomes attached to the messenger RNA, and then your tRNA will come here. There are several tRNAs for the different 20 amino acids. So one of the tRNAs will come here and look for complementarity with the mRNA sequence. So if there is complementarity, you have the first amino acid in place. And the next one will come here. Then there will be a bond formation, the peptide bond formation. And once this is attached, this will leave, go pick up another amino acid and come back and look for the matching triplet. So like that, you'll be adding more and more amino acids. And when you find the stop codon, one of the 3 stop codons, then no more amino acid will be attached. So that will be the end of the protein synthesis. So I want you to take a couple of minutes, look at figure 4.8 that shows all the steps. How the proteins are finally made and released into the ER. Steps 1 through 4. So step 1 shows mRNA coming out of the nuclear pore. Can you see that? And then you see the purple structures, which they call the ribosomes, going and attaching to the mRNA. And you see the tRNA also floating around, right? And if you see in step 2, that's a closer view of the ribosome and the mRNA. But you see there on the pink color, you see the mRNA codons, the triplets in the mRNA that code for the amino acids. And if a tRNA that's floating around with a certain amino acids, one of the 20 amino acids represented by the colored beads, if they find complementarity, they will stick there, meaning they will attach for a while the amino acid is in place. And there are 2 sites, okay? So one site, you'll have the first tRNA with amino acid at one end, and then the next amino acid will come in place. Then there will be a bond formation between the 2 amino acids, and the tRNA will leave. And then it keeps happening over and over and over again. So from the start to finish. So at a given time, if you see figure 3, you can see multiple ribosomes working on a single mRNA. Because if you use only one ribosome, it's going to take forever. You need maybe tons of proteins and in a short time, and it could be lots of different proteins you may need. So you'll have so many ribosomes working on the same mRNA at a given time. And then once the protein is synthesized, you can see the chain of beads is released into the ER in step 4. Can you see that inside? That's where it gets processed the first time. Then it may be going to the Golgi, where the Golgi apparatus will add more things if necessary, add some sugars and all if it has to be made into a glycoprotein, and then they will send it out. So all this happens in steps they call initiation, elongation, termination. So there is a starting point, then continuation of that, and then the termination. So you can see figure 4.9 actually what is happening in the cell. That shows you the ribosomes, the big dots, the black dots are your ribosomes, and in the background you see the loops, those are your mRNA and protein. So in the middle where the ribosomes are attached, you have the mRNA, whatever is looping out are the proteins. So this is a real-time image of what is happening. But figure 4.8 is just an illustration, but this shows actually what is going on. Many ribosomes processing a single mRNA strand.
Just out of curiosity, about how long does the whole process take to happen?
I don't know, micro nanoseconds or even less than that. Otherwise we cannot live. Our body, these enzymes and all of these, they are much more active than we are. Page 126, 410. This is a good example to summarize all of this that I was talking about. First you have the DNA double helix. Second you see the 7 triplets, which basically represent the code. But we call the actual code is on the mRNA, they call it the codon. So you see the complementarity there. You need to understand to know what is happening there. If you don't understand, you need to look up some YouTube video, or you can ask me, I can explain, but I cannot teach a whole class on this, but I'll help whatever extent I can. And then if you see step number 4, those are all your tRNAs corresponding to those codons. Okay? So already everything is predetermined in the first sequence, which is your triplet on the DNA. All we are doing is based on the complementarity you're selecting for something. So you have a blueprint before you build a house. Your house is decided when the engineer or the architect makes the blueprint, right? So the same way everything is in the DNA, only you are going through these steps before you make your final protein structure. This is a 6 amino acid long protein, what you have made there. You can see the 6 colored beads representing each different amino acid. And then figure 411 shows the different stages, how it moves once the protein is synthesized from the ER to the Golgi, and then it gets released. Some proteins may hang around in the cell, some may go to the membrane, because membrane, you remember, they are the protein channels. So they are also proteins. Your enzymes are proteins. There are many substances you need in different places. So some may stay in the cell, some may go out, some may stay in the membrane. So the vesicles, the primary function is storage and transport and secretion. So you may find transport vesicles, secretory vesicles, meaning transport vesicles simply transport, secretory vesicles bind to the membrane and release to the other side, secretion. And figure 4.12 shows gene activation, how a gene may be activated. Let's leave it alone for now. We will move on to DNA replication on the next page, figure 4.14. That shows you this first step. I told you the DNA makes copies of itself. That's called replication. So you can see there a single double-stranded DNA. It is opened up by one enzyme that's opening up the helix. So they call that enzyme DNA helicase. And once that's opened up, then there are other enzymes that arrange the nucleotides and make a new set of DNA. So you see the big green structure that's going and adding the new strand using the old strand as a template. So for every one single strand in the old molecule, you synthesize a new strand. So that means any DNA sequence has 50% the old copy. So half of it is conserved from the previous molecule. So you make only one new strand and add to it. And one strand, as you can see, is continuously synthesized, but if you see the second one, you have some gaps. There are some technical reasons for that, why that happens, and then those gaps are filled by another enzyme that is good at doing that, filling those gaps. Some enzymes can only work continuously, some come and fill in those gaps. Because DNA polymerase, there is not one enzyme, there are different forms of the same enzyme. The reason that we conserve one strand of the original DNA strand, we have two strands, one is conserved from the old parent molecule, they call it semi-conservative replication. And you can see the different enzymes, names: DNA helicase, DNA polymerase, DNA ligase, replication fork. Replication fork is the point at which you see the DNA is split and the replication is actually happening. It looks like a fork, right? And then you can see the helicase opening up the DNA strand and the polymerase following that and synthesizing the new strand. And then the ligase is the one that attaches these short pieces on the other strand. You see the short segments, the gaps, those are attached with the DNA ligase. So there are specific enzymes for each action. And coming to mutations, errors, so we have discussed that in the next section. Most of the mutations are rectified by enzymes that proofread once the new strand is made. If there is any error, then that nucleotide is fixed before the mutation is passed on. But in spite of all that, we have genetic mutations which we have no control. So that's where cancer is one such, depending on which part of the body you have. Cancer is nothing but there are genes that produce some products that control the cells from dividing without limit. They keep on dividing, but at some point they have to stop. But if they don't stop, that's when you have cancerous conditions. So it's just unlimited cell growth is what you call cancer. But of course they have all kinds of names for the different forms of cancer. Okay, that takes us to the cell cycle. The cell cycle involves a whole cycle of events that the cell goes through, including the different phases you have, the G1, S, G2, and the M phase in figure 415. So if your cell has to duplicate itself, now we are talking about the whole cell in addition to the DNA in the nucleus, okay? So it has to double all the contents, including the chromosomes. Okay, normally a human cell has 23 pairs of chromosomes. So if you have to make a daughter cell with the same number, so you have to first make it 46 pairs, then only you can split into two. And this goes on and on and on. So you double and duplicate, double and duplicate. This happens during that M phase, which is the mitotic phase. During the S phase, that's when the DNA duplication or replication occurs, so you double the chromosomes during the S phase. So what about the G1 and G2? The G1 and G2 are the gaps, or sometimes they call it a growth phase. That makes better sense than gap. That's the phase when the cell makes all the other components double. You need the mitochondria, you need more of everything, your Golgi, everything, so that you can equally share with the daughter cell. So S stands for synthesis, where DNA replication occurs, and M stands for mitosis, where you have the cell division. So cell division is part of the whole cell cycle, so you should not mix up cell division with cell cycle. So if you look in the M phase, there are again 4 phases within that, which is the prophase, metaphase, anaphase, and telophase. And you can see that better in page 133. You have an illustration that shows the 4 steps, figure 416. And also you can see the actual cell where you can see the division occurring. Prophase is where your chromosomes condense to become chromatids, and then the nuclear membrane surrounding that will disappear, or it will be degraded, and then you will have the centrioles on. If you see the cell, this is the nucleus, you will find the centrioles here. These centrioles, they will migrate to both ends, and then they will form these fibers they call spindle fibers. Each will attach to one of the, if you have the pair of chromatids, the centromere, they will attach from either side. And then in the metaphase, the spindle fibers move the pairs of chromatids to the mid plane of the cell. As you can see, they're all lined up in the middle. It's metaphase. And then the next phase is when the chromatid starts pulling away. That's your anaphase. And then finally, in the telophase, they start separating. And then your nuclear membrane forms again. Your chromatids again loosen up and become chromosomes like they were before. So you need to know the 4 phases and what happens in each phase. Where the chromatids are lined up in the middle, where it starts separating, where it starts completely dividing, when it starts to divide. So in order for the cell to divide, you have a structure called cleavage furrow. You can see under telophase, that's the membrane that folds inside and forms a furrow, and then it splits the content of the cell. So you end up with a cell that's similar to the parent cell with the same number of chromosomes. The division of the chromosome itself is called mitosis. The division of the cell contents is called cytokinesis. So there are 2 terms. Mitosis is more referring to the nuclear or chromosomal division, but the cytoplasm also is divided, shared between the 2 cells. They call it cytokinesis. So when your cell splits into 2, the structure that helps to do that is the cleavage furrow, and the division of the cytoplasm is called cytokinesis. That occurs at the time of telophase. Okay, the last part, it talks about chromosomes and some of the defects. I want to give you an introduction, but next class we will finish it up. What you see there is a display of the various chromosomes that human cells have, or any organism. You can take the chromosomes out, and there is a way the experts can arrange the chromosomes and look at them and match them in pairs. They call this karyotype. So everybody has a karyotype. Like our fingerprint, we have a genetic fingerprint too, which is the 23 pairs of chromosomes. That's what called the karyotype. Based on that, see some differences can be figured out in terms of genetic defects, or if your chromosome is missing, or maybe one chromosome is extra. All those lead to genetic disorders. You have 23 pairs of chromosomes. The last one, the pair number 23, that has the XY, those are the sex chromosomes because they determine whether it's a male or a female. So XX will be a female, XY will be a male. As you can see, the Y is a little shorter than the X chromosome. And then you see those words, haploid, diploid. Haploid has half the number of chromosomes. If you have the complete 23 pairs, it's a diploid. If you have only one set, it's called haploid. So your sperms and eggs, they have haploid chromosomes. They don't have the pair, because otherwise you'll have twice the number of chromosomes in the baby, which you don't want. Then you have germ cells and somatic cells. Germ cells are the cells that become sperms and eggs. Somatic cells are the ones that develop into other types of tissue. Then you can see the alleles, locus, dominant, recessive. Locus is simply the location your genes are located in a certain place in your whole DNA sequence. And alleles are the mirror images of one and the other. Like if you have one from male, one from female, you have a pair, right? Pair of chromosomes. They call them alleles. One came from male, one came from female. And sometimes one is dominant over the other. Sometimes one is recessive. Dominant means as long as one copy of the gene is there, it will dominate that characteristic or that trait. Maybe certain color of your eye or color of your skin or whatever. The dominant allele and the recessive allele can only express when the dominant allele is not expressed. Then they have discussed genotype, phenotype. There are some more terms, homozygous, heterozygous. So we will have to go through all of that and look at some of the problems associated with the chromosome disorders. Okay. And we will discuss a little bit of the tissue, chapter 5. We will not do the skeletal tissue, muscle and nervous tissue. We will do as we discuss about those muscles and bones and brain and all of that. But we will get started on chapter 5. So we'll finish chapter 4 and we will start with chapter 5, the introductory part where they discuss the epithelial tissue and connective tissue. Okay.